Welcome to Biology at Priyojit, my dear viewers. Today we explore an essential topic in cell biology, that is cell junctions. Just as bricks are held together to build a strong wall, cells are connected by specialized structures called cell junctions. These junctions not only hold cells together but also allow communication and coordination between them. From sealing spaces between cells to providing mechanical strength and enabling direct exchange of signals, occluding, anchoring, and communicating junctions play a vital role in maintaining tissue structure and function. So let's understand these junctions in a simple and systematic way. Cell junctions, basically they are specialized structures that connect neighboring cells or attach cells to the extracellular matrix or ECM. Cell junctions are essential for maintaining tissue integrity, cell-to-cell communication, proper coordination and functioning of cells in the tissues. So classification of cell junction. Cell junction can be broadly classified into three main types: occluding junctions, anchoring junctions and communicating junctions. So types of junctions, there are tight junctions, anchoring junctions and there are gap junctions. So basically there are three types of junctions: occluding, anchoring and communicating junctions. Occluding junctions, they seal the intercellular space between adjacent cells. They prevent free movement of substance across epithelial layers. So one example of occluding junction is the tight junction, which is also known as zonula occludens. They are found mainly in epithelial tissue. This is the epithelial tissue, and in between cells of the epithelial tissue there are tight junctions. They are located at the apical region of the cell and they form a continuous belt-like seal around the cell. Its function is to prevent leakage of molecules between cells or paracellular transport. It maintains cell polarity by separating apical surface and basal surface. It regulates selective permeability of epithelial layers. Proteins involved: claudins, occludins and junctional adhesion molecules. Septate junction or occurrence and structure. It is common in invertebrate epithelial cells such as insects. Is located below tight junctions. It possesses a ladder-like or septum-like structure. So functions: it provides a strong barrier similar to tight junctions. It maintains tissue integrity and it controls permeability. This is the image showing the septate junction. Anchoring junction. Anchoring junctions basically provide mechanical attachment. This is the image showing the anchoring junction, which give strength and stability to tissues. They connect cell to cell or cell to ECM. And types of anchoring junctions, there are two major types. Number one is cell-cell anchoring junctions, and it includes adherens junctions or zonula adherens, which form a belt-like structure located just below the tight junction, and it connect actin filament of the two adjacent cells. Proteins involved in is cadherins, which is calcium dependent adherens junctions, and catenins. They are examples of the adherens junctions and adherens junction proteins. So they maintain tissue shape and they play important role in embryonic development and cell movement. Desmosomes or macula adherens structure, they appear as spot-like junctions. They connect intermediate filaments of the neighboring cells. Here the proteins involved are desmoglein and desmocolin, which are also known as cadherins, and desmoplakin. So functions include: it provides strong cell adhesion, it prevents cells from pulling apart, and it commonly found in skin and cardiac muscle. So this is the image showing the structure of desmosome, and this is the image of adherens junction. This is the types of anchoring junctions. There is adhering junctions and desmosomes, hemidesmosomes, etc. Cell matrix anchoring junctions. There are two types of anchoring junctions. One is cell to cell, which we have already studied, and now we will study about the cell matrix anchoring junctions. The first example of cell matrix anchoring junctions are the hemidesmosomes. They anchor epithelial cells to the basement membrane. They connect intermediate filaments to the extracellular matrix. Integrin and collagen 17 are the proteins involved in hemidesmosome formation, and it provides stable attachment of epithelial cells to the underlying tissues. Focal adhesion or structure. The focal adhesion is another type of cell matrix anchoring junction, and its structure include attached actin filaments to the ECM, and it is a highly dynamic junctions. Functions: it facilitates cell migration, it is involved in signal transduction, and important for tissue remodeling. This is the image showing the focal adhesion. Communicating junction. These junctions allow direct communication between adjacent cells, such as gap junctions. Gap junctions are basically made up of protein channel known as connections. Each connection consists of connexin protein, and its function: it allows passage of ions, metabolites, small molecules. It enables electric coupling and metabolic coupling. Especially it is important in cardiac muscle, smooth muscle, and embryonic tissues. Conclusion: so tight junction is responsible for sealing, adherens junction is act as like actin, desmosome is made up of intermediate filament, hemidesmosome is attached with the basement membrane, and gap junction function is communication. So as we conclude, remember that cell junctions are the silent architects of tissues organization, by sealing spaces, providing mechanical strength, and enabling direct communication. Occluding, anchoring, and communicating junctions work together to maintain cellular harmony. Understanding these connections is not just exam oriented, it helps us appreciate how cells function as a coordinated unit rather than in isolation. With this clarity, you are now better prepared for deeper studies in cell biology. So keep revising, stay curious, and continue exploring the fascinating connections with the cellular world. Thank you.